Okay, so welcome to week 2 of XViz. This week we're going to be doing bioenergetics for the whole week. And to start off, we're going to talk about substrates, or basically things that we use to help create energy, and then a little bit about energy production. So here are the learning topics and outcomes for this particular lecture. So to start off, what are the primary energy substrates? In other words, where do we get energy from? And those, of course, are carbohydrates, proteins, and fats. Okay, when we think of carbs, we think of sugars. That's essentially what they are. There's several different types of sugars, and you can get into complex carbohydrates and things like that. But when it comes down to it, we're looking for sugar. We're using the sugar out of carbohydrates. So the one that we talk about most often with bioenergetics, and that we'll be talking about later on this week, is glucose. And glucose is just a simple sugar. It's what we call a monosaccharide, and it's composed of 6 carbohydrates. Now, think of glucose as a one link in a chain. If we put a lot of links together, that is essentially how we form glycogen. And glycogen is how we store, or is the storage form of glucose, and this is stored in the liver. Okay, so if we've got low blood glucose, what happens is the hormone glucagon is released. Glucagon goes to the liver and basically takes glycogen and breaks it up into its individual glucose molecules. When it comes to nutrition, and we'll talk a little bit more about this as we go throughout the semester, we talk about calories, things like that. 1 gram of carbohydrates equals 4 kilocalories. Okay, so remember that 1 gram of carbohydrates, 4 kilocalories. Next, we've got fats. And there's, again, several different types of fats. We'll dive into those more in detail later. But for right now, just kind of want to focus big picture. So when it comes to fats, we primarily are looking at triglycerides. Those are what we use for energy. And so as it says here, a triglyceride is a glycerol molecule, or there's one glycerol molecule that has 3 fatty acids attached to it. So you could just think of something like here's my glycerol molecule, and then these are fatty acid chains. And they can be in varying lengths. And so there you go. And what we do is we break the fatty acid chains off the glycerol, and then we break the fatty acid chains down into into pairs of carbons that we can do other things with, that we'll talk about on Thursday of this week. When we're not using fat, they're stored in adipose tissue. Fats actually hold a lot of energy. And so 1 gram of fats, we get about 9 kilocalories. So more than double what we get from carbohydrates. Next, we've got proteins, and proteins are just chains of amino acids. And amino acids are these compounds that contain nitrogen. There's a lot of different types of proteins that we'll talk about more next week. But they're a relatively small source of energy. Okay, we don't really use protein for energy that much. We use protein for other things. And in fact, in order to use proteins, we generally convert them into glucose or into fat. And so these processes are referred to as gluconeogenesis as well as lipogenesis. Okay, genesis means formation or beginning, so we're creating or forming glucose, or creating or forming fats. When we do use these proteins for energy, we get 1 gram of protein equals 4 kilocalories. Okay, now when it's time for us to produce energy, or produce compounds that we can use energy, when it comes to this energy, we want to control when we produce it. Okay, we don't want it just to be produced willy-nilly, and we want to be able to break it down when we need it, that sort of thing. And so, in order to do this, or what kind of limits this or determines the rate, it's probably a better word, like it says here, is the availability of the substrate. So how many carbs, proteins, fats do we have? And then do we have the necessary enzymes? Okay, enzymes help speed things up, make it easier for chemical reactions to occur. So in order for us to take a molecule of glucose and form ATP from it, we need to be able to have the enzymes that'll take that glucose through 10 steps of glycolysis or 8 steps of Krebs, and so on and so forth. Okay, so generally the way it works is if I've got a lot of a substrate, so if I've got a lot of glucose, then I'm going to have a greater ability to generate energy from glucose. All right, and like I said just a second ago, enzymes speed up these chemical reactions. What they do is they lower the threshold, like it says here. And what we mean by that is that for chemical reactions to occur, there has to be energy. Okay, and so what this picture is showing here is that let's say we've got phosphocreatine and we want a reaction to occur. Well, we've got to get up to this point. Okay, it's called activation energy, and we're not going to dive into that, but just think of it's just kind of what you need to start the car. Okay, and so we need a relatively large amount, but if we've got creatine kinase, the amount of energy required to start that reaction is reduced. And so you could look here, it's reduced by, a third or half or something like that. And so enzymes are important because they make it easier for these reactions to occur and they speed them up. Okay, but in order for some of these enzymes to work, they need what's called a cofactor. And I think of cofactors and enzymes kind of like what's going on here. So here we've got a safety deposit box, and it's not a perfect analogy, but we've got a safety deposit box. In order for someone to get into the safety deposit box, you have to have 2 keys. And so just think of the enzyme as one of those keys, and then the cofactor is another key. Okay, if I don't have this key here, so we're just going to say this person isn't there. Well, I can have the enzyme present and I can have lots of it present, but if I don't have this other key, that enzyme's essentially useless. Okay, and so we're going to end right there.